Bismillahirrahmanirrahim. Today's my lecture is about primary active transport. At first, active transport. Cells cannot rely solely on passive transport of substance across their membrane. In many instances, it is necessary to move substance against their electrical or chemical or electrochemical gradient to maintain their appropriate concentration inside the cells or organelles. At times, a large concentration of a substance is required in the intracellular fluid, even though the extracellular fluid contains only a small concentration. This situation is true for potassium ion. Conversely, it is important to keep the concentration of other ion very low inside the cell, even though their concentration in the extracellular fluid are great. This situation is especially true for sodium ion. Neither of these two effects could occur by simple diffusion because simple diffusion eventually equilibrates the concentration on two sides of the membrane. Instead, some energy source must cause excess movement of potassium ion to the inside of the cell and excess movement of sodium ion to the outside of the cell. When a cell membrane moves molecules or ion uphill against a concentration gradient, this process is called active transport mechanism or active transport process. So, what is active transport? Active transport is the movement of ion or substance across a membrane against the chemical or electrical or electrochemical gradient with a carrier protein which require energy. This energy is obtained mainly by breakdown of high energy compound like adenosine triphosphate. It is also called uphill transport. It is like swimming against the water tide in a river. Mechanism of active transport. When a substance to be transported across the cell membrane comes near the cell, it combines with the carrier protein of cell membrane and forms substance protein complex. This complex moves towards the inner surface of the cell membrane. Now the substance is released from carrier protein. The same carrier protein moves back to the outer surface of the cell membrane to transport another molecule of the substance. This is the figure for active transport process. Active transport is divided into two types according to the source of energy used to facilitate that transport. That is primary active transport and secondary active transport. In primary active transport, the energy is derived directly from breakdown of adenosine triphosphate or some other high energy phosphate compound. And in secondary active transport, the energy derived secondarily from energy that has been stored in the form of ionic concentration difference of secondary molecular or ionic substance between the two sides of the membrane, created originally by primary active transport. In both instances, transport depends on carrier protein that penetrate through the cell membrane, as it is true for facilitated diffusion. However, in active transport, the carrier protein functions differently from the carrier protein in facilitated diffusion because it is capable of imparting energy to the transported substance to move against electrochemical gradient. Active transport needed for maintaining chemical and electrical charge at rest. Intake of substance through gated channel. Thirdly, collecting of ions with more concentration. Substances which are transported actively are in ionic form and non-ionic form. In ionic form, these are sodium, potassium, calcium, hydrogen, chloride and iodide. And non-ionic form are glucose, amino acid and urea. The pump involved in active transport in many cells, it is sodium-potassium pump, and in membrane of sarcoplasmic reticulum it is calcium pump, and in gastrointestinal cell membrane these are potassium hydrogen pump. At first, primary active transport of calcium ion. Calcium ion are normally maintained at an extremely low concentration in intracellular cytosol of virtually all cells in the body at a concentration of about 10,000 times less than in the extracellular fluid. This level of maintenance is achieved mainly by two primary active transport of calcium pump. One, which is in the cell membrane, pumps calcium to the outside of the cell. The other pump calcium ion into one or more of the intracellular vesicular organelles of cell, such as the sarcoplasmic reticulum of muscle cell and mitochondria in all cells. In each of these instances, the carrier protein penetrates the membrane and functions as an enzyme ATPase with the same capability to cleave ATP as the ATPase of the sodium carrier protein. The difference is that this protein has a highly specific binding site for calcium instead for sodium. Primary active transport of hydrogen ion. Hydrogen ion is actively transported across the cell membrane by carrier protein called hydrogen pump. It also obtains energy from ATP by activity of ATPase. The hydrogen pump that is present in two important organs have some functional significance. In stomach, hydrogen pump in parietal cell of the gastric gland are involved in the formation of hydrochloric acid, and in kidney, hydrogen pump in epithelial cell of late distal convoluted tubule and cortical collecting duct are involved in the secretion of hydrogen ion from blood into urine. At first, in the gastric gland, the deep-lying parietal cell have most potent primary active mechanism for transporting hydrogen ion of many parts of the body. This mechanism is basis for secreting hydrochloric acid in stomach gastric secretion. At the secretory end of the gastric gland parietal cell, the hydrogen ion concentration is increased as much as million-fold and then is released into stomach along with chloride ion to form hydrochloric acid. And in renal tubule, special intercalated cells found in the late distal tubule and cortical collecting duct also transport hydrogen ion by primary active transport. In this case, large amount of hydrogen ion are secreted from blood into urine for the purpose of eliminating excess hydrogen ion from the body fluid. The hydrogen ion can be secreted into the urine against a concentration gradient of about 900-fold. Then most important pump is sodium-potassium pump. Sodium-potassium pump is a transport process that pumps sodium ion outwards through the cell membrane of the all cell and at the same time pumps potassium ion from outside to inside. This is also called sodium-potassium ATPase pump or sodium-potassium ATPase. Driven by splitting of ATP to provide energy and conformational changes of proteins by adding and then take away a phosphate group. This pump is present in all cells of the body. Here, 3 sodium ion moves out of the cell and then 2 sodium ion moves into the cell and used to establish an electrochemical gradient. This pump is responsible for maintaining sodium and potassium concentration difference across the cell membrane as well as establishing a negative electrical voltage inside the cells and developments of resting membrane potential. Indeed, this pump is the basis for nerve function, transmitting nerve signal throughout the nervous system. In this lecture, we mostly elaborately discuss about the sodium potassium pump, because this pump is very much important for our body and for our education. In mechanism of actions of sodium potassium pump, 3 sodium ion from cell get attached to the receptor site of sodium ions in the inner surface of carrier protein, and 2 potassium ion outside the cell binds to the receptor sites of potassium ion located on the outer surface of carrier protein. Bindings of sodium and potassium ion to the carrier protein activate the enzyme ATPase. ATPase causes breakdown of ATP to adenosine diphosphate and release one high energy phosphate. This is the hypothetical diagram of sodium potassium pump. Now, the energy liberated causes some sort of conformational change in the molecules of carrier protein. Because of this, the outer surface of the molecules now faces the inner side of the cell, and the inner surface of the protein molecules faces the outer side of the cell. Dissociation and release of the ion takes place, so that the sodium ion are released outside the cell and potassium ion are released inside the cell. Okay. Then we elaborately discuss about the carrier protein for sodium potassium pump. Structure of sodium potassium pump carrier protein that constitutes sodium potassium pump is made up of 2 separate globular proteins. A large one called alpha subunit with a molecular weight is about 1 lakh, and a smaller one is called beta subunit, which is a glycoprotein. Its molecular weight is about 55,000. Although the functions of smaller protein is not known, except that it might anchor the protein complex in lipid membrane. The larger protein has 3 specific features, and alpha subunit of sodium potassium pump has got 6 sites. At first, it has 3 binding sites for sodium ion on the portion of the protein that protrude to the inside of the cell, and it has 2 binding sites for the potassium ion on the outside. The inside portion of this protein near the sodium binding sites has adenosine triphosphatase activity. When 2 potassium ion binds on the outer sides of the carrier protein and 3 sodium ion binds on the inside, the ATPase function of the protein becomes activated. Activation of ATPase functions leads to cleavage of one molecule of ATP, splitting it to adenosine diphosphate and liberating high energy phosphate bound of energy. This liberated energy is then believed to cause a chemical and conformational changes in this protein carrier molecule, and extruding 3 sodium ion to the outside and 2 potassium ion to the inside. This is the figure which postulated the mechanism of sodium potassium pump. As with other enzymes, the sodium potassium ATPase pump can run in reverse. If the electrochemical gradient for sodium potassium are experimentally increased to the degree that energy stored in their gradient is greater than the chemical energy of ATP hydrolysis, these ions will move down their concentration gradient, and sodium potassium pump will synthesize ATP from ADP and phosphate. The phosphorylated form of sodium potassium pump therefore can either donate its phosphate to ADP to produce ATP, or use the energy to change its conformation and pump sodium ion out of the cells and potassium ion into the cell. The relative concentration of ATP, ADP and phosphate as well as electrochemical gradient for sodium and potassium determine the direction of the enzyme reaction. For some cells, such as electrically active nerve cell, 60 to 70% of the cell's energy requirement may be devoted to pumping sodium out of the cell and potassium into the cell. What is electrogenic nature of sodium-potassium pump? The answer is the fact that sodium-potassium pump move 3 sodium ion to the exterior for every 2 potassium ion that are moved to the interior. Means that a net of 1 positive charge is moved from the interior of the cell to the exterior for each cycle of pump. Continuous activity of sodium-potassium pump causes positivity outside of the cell, but reduction in the number of positivity charged ion inside the cell, leading to increase the negativity inside the cell. Therefore, sodium-potassium pump is said to be electrogenic because it creates an electrical potential across the cell membrane. This electrical potential is a basic requirement in nerve and muscle fiber for transmitting nerve and muscle signal. In inhibition of the sodium-potassium pump, the pump requires binding of sodium and potassium and ATP for its operation. If the concentration of any of these is low, the functions of the pump is low. Decreases temperature, oxygen lack, metabolic poisons is also responsible for inhibition of the pump. It's also by cardiac glycosides dopamine, which inhibits the pump. The sodium-potassium pump activity is increased by aldosterone, thyroid hormone, and insulin. Importance of sodium-potassium pump in physiological basis: responsible for generation of membrane potential, control the cell volume. Example is maintain the red blood cell volume. Basis of nerve and muscle functions, that means maintaining the high potassium and low sodium concentration inside the cell, and provide energy for secondary active transport. In clinical importance, sodium-potassium pump inhibitor is used in heart failure and a diuretic which inhibit the sodium-potassium pump, which causes natriuresis. One of the most important functions of sodium-potassium pump is to control the volume of cell. Let's discuss about it. Without the functions of this pump, most cells of the body would swell until they burst. The mechanism for controlling the volume are discussed elaborately. Inside the cell, there are large number of proteins and other organic molecules that cannot escape from the cell. Most of these protein and other organic molecules are negatively charged and therefore attract large number of potassium, sodium, and other positive ion as well. All these molecules and ions then causes osmosis of water to the interior of cell. Unless this process is checked, the cell will swell indefinitely until it bursts. The normal mechanism for preventing this outcome is sodium-potassium pump. Also, the membrane is far less permeable to sodium ion than to the potassium ion. Thus, once sodium ions are on the outside, they have strong tendency to stay there. This process thus represents a net loss of ions out of the cell, which initiates osmosis of water out of the cell as well. If a cell begins to swell for any reason, the sodium-potassium pump is automatically activated and moving still more ions to the exterior and carrying water with them. Therefore, the sodium-potassium pump performs a continual surveillance role in maintaining the normal cell volume. Abnormalities of the sodium-potassium pump, which are associated with several pathological conditions. Examples are Reductions in either number or concentration of sodium-potassium pump in myocardium is associated with cardiac failure. And excess reabsorption of sodium in renal tubules is associated with hypertension. And ion channel disease or channelopathies, which are caused by mutation in genes that encode in ion channel. At first, sodium channel disease. Dysfunctions of sodium channel disease to muscle spasm and Liddle's syndrome. In Liddle's syndrome, dysfunction of sodium channel in kidney resulting increased osmotic pressure in the blood and hypertension. In potassium channel dysfunction causes disorder of heart, inherited deafness and epileptic seizure in newborn. Chloride channel disease, dysfunction of chloride channel results in formation of renal stone, cystic fibrosis. Cystic fibrosis is a generalized disorder affecting the functions of many organs such as lungs, exocrine gland like pancreas, biliary system, immune system. Thank you for your patience hearing.